Hello, everyone. Thank you so much for joining. My name is Olivia Graham, and I'm excited to share with you some of my work exploring how marine herbivores influence the transmission of a seagrass pathogen. Before getting into my research, I first would like to thank my many colleagues and collaborators who helped support this work and really made it possible, including a number of terrific undergrads. Small herbivores can have big ecological impacts, especially when they work in tandem with plant pathogens, as we've seen with barley yellow dwarf virus and Dutch elm disease. While there's a large body of work on terrestrial vector-borne plant pathogens, there isn't really for marine plant pathogens. This is a little funny because seagrass meadows are like grassy fields, but underwater, with lots of grass, an infectious pathogen, and hungry herbivores. Still, there currently are not any reports of disease vectors in seagrass meadows, which I study. I study seagrass wasting disease caused by the marine protist Labyrinthula zostera. I'll refer to it in this talk as LABI. LABI creates these dark necrotic lesions, which you can see here, and it can kill plant tissue. This pathogen can also reduce leaf growth and below ground sugar reserves. Altogether, this is an ecologically, economically significant disease. Common eelgrass herbivores include gastropods, such as snails, as well as arthropods, including amphipods and isopods. Studying herbivore interactions with diseased eelgrass is important because currently there are only two known modes of disease transmission. That is via direct contact between an infected and healthy eelgrass leaf and through seawater. Herbivore-facilitated disease transmission could have big impacts on our understanding of the spread of this disease. For this project, we had several research questions, the first of which was, can herbivores increase disease? Here, I hypothesized that herbivores can indirectly facilitate disease transmission by either creating grazing scars through which the pathogen could enter and infect eelgrass, or through their feces, which could contain infectious pathogenic cells. For my second question, I wanted to explore if herbivores preferentially feed on diseased eelgrass. And here I expected that some herbivores prefer to consume diseased over healthy eelgrass. Finally, I wanted to address the question of if herbivores that feed on diseased eelgrass contain the pathogen. And here I expected that herbivores that feed on diseased eelgrass would test positive via qPCR for the pathogen Labyrinthula zostera. To get at these questions, I conducted a series of herbivore experiments whereby I tested isopods and snails, fed them healthy eelgrass, allowed them to create nice grazing scars, and then I inoculated with either concentrated pathogen or sterile seawater. I then incubated these blades and allowed lesions to develop, at which point I collected the eelgrass blades, scanned them, and measured wasting disease and herbivory. I did not test amphipods here because when I tried to feed them healthy eelgrass tissue, they would not consume it. I should also note that these trials test for indirect disease transmission. And that is because earlier trials where I tested for direct transmission of the pathogen seemed that that is an unlikely mechanism of disease spread. Getting into some of our results, here we are looking at disease severity, that is the proportion of infected tissue on a given eelgrass leaf across our different treatments with and without herbivores and our controls that we inoculated with water. And we can see in the treatments with isopods and snails the same result where our eelgrass with herbivores and inoculated with the pathogen had greater disease severity compared to those eelgrass without herbivores, but that were also inoculated. Likewise, when we look at the consumed leaf area plotted against the disease severity, we can see plants that had greater grazing scars, greater tissue loss, also had greater disease severity, indicating that those grazing scars are likely important in disease transmission. Briefly, we also found that herbivores had different herbivory preferences, with amphipods preferring diseased tissue, while isopods and snails preferred to consume healthy eelgrass. Our qPCR analyses indicated that the pathogen was in fact detectable in herbivores that consumed diseased eelgrass, whereas it was not detectable in herbivores that consumed healthy eelgrass. And with that, I would like to take Any questions? Feel free to reach out. Thank you.